This video is presented by Subi, lecturer from Emmanuel Arasar College of Pharmacy. Today I am going to teaching about tablets, different tablets. Tablets is a solid dosage form containing medicinal substance with or without suitable diluents, prepared either by compression or molding method. They may be either circular, flat or biconvex in shape. Tablets is one of the most popular pharmaceutical dosage form. Next, move on advantages of tablets. First, tablets are unit dosage form with the excellent dose precision and least content variability. Yes, tablet is most accurate dosage form than other dosage form. Next, they are stable, elegant, effective and convenient dosage forms. Tablets have more chemical and microbial stability compared to other dosage forms. They are economical compared to other oral dosage forms. These tablets are suitable for large-scale productions. They are easy to handle, cheap to transport and package. They can be formulated as sustained or controlled release formulation. Active pharmaceutical ingredients with a bitter taste and unpleasant odor can be masked by using suitable coating techniques. This is the most important advantages of the tablets. They are tamper-proof dosage forms. Next, move on disadvantages of tablets. Tablets are very difficult to swallow, especially by pediatric, elderly and unconscious patients. Drugs which are microscopic or amorphous in nature or having low density are difficult to compress into dense compacts. Drugs with poor wetting and slow dissolution properties are not suitable for tablet formulation, which may exhibit poor bioavailability. Drugs which have bitter taste or sensitive to moisture or oxygen will require special type coating, which may increase the cost of production. Next, move on classification of tablets. Tablets are classified into 4 different types. They are tablet ingested orally, tablets used in oral cavity, tablets administered by other route, tablets used to prepare solution. Then first one, tablet ingested orally. These tablets again classified into 8 types. They are, first one is compressed tablet, second is multiple compressed tablet, third one is swivel tablets, fourth one is sugar coated tablets, fifth one is film coated tablets, sixth one is delayed release tablet, seventh one is controlled release tablet, and eighth one is sustained release tablets. Again, tablets used in oral cavity divided into four types. They are lozenges and troches, buccal and sublingual tablets, dental cones, mouth dissolving tablets, or fast dissolving tablets. Tablet administered by other route, vaginal tablets and implantation tablets. Tablets used to prepare solution. They are classified into four types. They are effervescent tablets, dispensing tablets, hypodermic tablets, and tablet trichrates. First see about compressor tablet. These are uncoated tablets made by compression of granules. They provide rapid disintegration and drug release. Example, paracetamol tablets. Next, multiple compressor tablets. These are layered tablets composed of at least two layers. These two layers tablet used to separate physically or chemically incompatible drugs to produce repeat action or prolonged action products. What is mean by repeat action? Repeat action means tablet release initial dose followed by second dose. What is mean by prolonged action? Release of medication is slowly. These tablets made in multiple compressed tablet method. Example, clomipiride and metformin bilayer tablets. These tablets one of the diabetic tablets. Next, chewable tablets. These tablets to be chewed in the mouth before swallowing. Example, antacid tablets, that means gelisil tablets, and vitamin tablets, means linseed tablets. Next, move on, sugar or chocolate coated tablets. These are compressed tablets with sugar or chocolate coating. Because of some tablets having unpleasant taste, so we have used sugar or chocolate coating to inhibit unpleasant taste and also to improve the stability of the drug. Example, multivitamin tablets. Next, move on, film coated tablets. These are compressed tablets with the coating on the surface using film forming polymers. Film forming polymers such as chitosyl cellulose, ethyl cellulose, chitosyl propyl methyl cellulose, etc. Film coating is done to improve the appearance of the formulation, mask the bitter taste and protect the formulation from atmospheric effects. Example, ibuprofen film coated tablets. Next, delayed release entry coated tablets. These are compressed tablets coated with enteric polymers such as cellulose acetate phthalate, which does not disintegrate in stomach but dissolve in occurring pH in the intestine. This tablet does not dissolve in stomach in acidic pH, but it is dissolved in alkaline pH of the intestine. Example, rabiprazole tablet. This is one of the anti-ulcer tablet. Next, move on. Release tablets. These are the tablets intended to release the drug in the stomach slowly at a predetermined rate for a certain time period after ingestion and successful in maintaining constant drug levels in the blood or target tissue. Example, paroxetine hydrochloride CR tablets. Next, move on. Sustained release tablets. These tablets are prepared using sustained release polymers, such as which release the medicament for prolonged period of time, thereby minimize the frequent administration of drugs. Example, diclofenac XR tablets. Next, move on. Tablet used in oral cavity. First, lozenges and torches. These tablets are designed to exert a local effect on mouth or throat. Means these tablets are used to treat sore throat or to control cough. Lozenges are generally made with the drug incorporated in the flavored hard candy sugar base by molding method or compression. Example, strepsils or clotrimazole torches. Next, buccal and sublingual tablets. Buccal tablets are placed in buccal pouch or between the teeth and cheek. Sublingual tablets are placed under the tongue. These tablets are intended to be held within the oral cavity where they dissolve, disintegrate quickly and absorbed directly into the blood vessels of the oral mucosa without passing into the alimentary canal. Example, hormonal tablets, isosorbide dinitrate tablets. Next, dental cones. These are the tablets used to place in the empty sockets, means remaining after tooth extraction, used to prevent the growth of bacteria in the socket. Example, parasop cone. Next, mouth dissolving tablets or fast dissolving tablets. They are designed to dissolve in the drug in saliva within few seconds. Super disintegrants are used in this formulation to enhance the fast disintegration of tablets in the buccal cavity. Example, aceclofenac tablets. Next, we move on. Tablets administered by other route. Vaginal tablets. These tablets are designed to use in the vaginal cavity. They are used to treat vaginal infections. Example, clotrimazole vaginal tablets. Next, implantation tablets. These tablets are placed under the skin by means of minor surgery. These tablets are slowly absorbed. They are capable of releasing the drug for prolonged period of time from 3 to 6 months. They are capable of releasing the drug for prolonged period of time from 3 to 6 months. Example, testosterone tablets. Then tablets used to prepare solution. First one is effervescent tablets. These tablets, when added in water, produce effervescence due to evaporation of carbon dioxide. They are designed to dissolve rapidly in water due to the chemical reaction which take place between alkali, bicarbonate and acid. Acid means citric acid or tartaric acid. This flavored carbonates didn't help to mask the unpleasant taste of some drugs. Example, Disprin tablets. These tablets is one of the effervescent tablets due to evaporation of carbon dioxide. Next, move on dispensing tablet. These tablets are intended to be added to the given volume of water before administration, which contain super disintegrants that quickly dissolve the drug to form a clear solution. Example, Digiplex. Digiplex is one of the enzyme tablets. Next, hypodermic tablets. These tablets are used by physicians for the extemporaneous preparation of the parenteral solutions. These tablets are dissolved in sterile water and administered parenterally. Example, Hyosin tablets. Tablet triturates. These are very small, cylindrical, molded or compressed tablets containing potent drug with a suitable diluent. Example, enzyme tablets. Next we see about formulation of tablets. In addition to API, full form of API is active pharmaceutical ingredients. Tablet contains a number of pharmacologically inert materials known as additives or excipients. Each excipient has its role in the tablet formulation. Tablet excipients. First one is filler or diluents. Second one is disintegrants. Third one is binders or adhesives or granulating agents. Fourth one is glidants. Fifth one is lubricants. Sixth one is anti-adherents. Seventh one is organoleptic additives. First see about fillers or diluents. These are excipients used in the tablet to increase the bulkiness of tablet when the proportion of medicament itself is inadequate to produce the bulk. If we have sometimes prepared tablet, he does not give adequate bulkiness. So we have added some fillers. It's used to increase the bulk of tablets. The addition of diluent provides better tablet properties such as improved cohesion, promote flow, and aid in easy compression of tablets. Fillers is mainly used to increase bulkiness of tablets. It is also used to improve cohesion, promote flow, and easy compression of tablets. If the dose is very high, no need of filler. Examples: lactose, sucrose, starch, mannitol, sorbitol, dibasic calcium phosphate dihydrate, dextrose, microcrystalline cellulose, etc. These are the most important used of fillers used in tablets formulation. Next, move on. Second one is disintegrants. What is mean by disintegrants? Means solid particles, large solid particles converted into small large particles converted to smaller particles. They are used in the tablet formulation to facilitate breakup of tablets when it contacts with the water and GIT. GIT means gastrointestinal tract. For example, starch, cellulose, cross-linked polyvinyl pyrrolidone, microcrystalline cellulose, sodium carboxymethyl cellulose, etc. Disintegrants are commonly used for the rapid disintegration of tablets such as oral dispersible tablets, fast dissolving tablets, etc. Example, sodium starch glycolate, cross-carbamylose sodium, cross-povidone. Next we move on. Binders or adhesives or granulating agents. Binders are added to improve the adhesive property of compressing materials. They provide moisture to convert the fine powder into damp mass, which after passing through a sieve forms granules. They hold the ingredients of the tablet together and provide mechanical strength to the tablets during and after compression. For examples, polyvinyl pyrrolidone, starch derivatives, alginates, cellulose derivatives, etc. Next we move on glidants. They are used to promote the flow of the granules or powder materials by reducing friction between the particles or granules. Talc, cornstarch, colloidal silica or aerosol is mostly important used in glidants. Next, see about lubricants. Lubricants, they are used to reduce the friction during tablet ejection between the walls of tablets and the die cavity in which the tablet was formed. Example, polyethylene glycols, stearic acid derivatives, talc, etc. And adherents. They have both the function of lubricants and also glidants. They are used to prevent sticking or adhesion of the tablet granulating materials or powder to the face of punches or to the die wall. Colloidal silica, cornstarch, magnesium stearate, calcium stearate, stearic acid, talc. These are the all of active SCM and the adherents. Next move on organoleptic additives. Colors, flavors and sweetness are the organoleptic additives used in tablet formulations. First one is colors. Colors is the most important organoleptic additives in tablet formulation. The purpose of using colors in the tablet formulation is to discuss of color drugs, product identification, and to develop a more elegant product. All coloring agents must be approved and certified by FDA. FDNC and DNC dyes and lakes are the two forms of approved colors that have been used in tablet formulations. Examples: Yosin red, sunset yellow, tartrazine yellow, brilliant blue, erythrocin, etc. These are the examples of colors used in tablet formulation. Next we move on flavors. Flavors are Flavors are used to produce a pleasant smell or to mask unfavorable odor. They are important in case of chewable tablets or tablets intended to dissolve in mouth. Example: fruit or citrus flavors are frequently used to combat sore, air acid test in drugs, and cinnamon. Orange, raspberry, and other flavors have been successfully used to make preparations of salty drugs more palatable. Finally, we have seen about sweeteners. Sweeteners is one of the most important additives in tablet formulation because they inhibit unpleasant taste and odor. They are used in chewable tablets and mouth dissolving tablets to mask the bitterness. They improve the palatability, hence enhances the uh, acceptability of tablets. Examples: mannitol, aspartame. It is an artificial non-saccharid sweetener, 180 to 200 times sweeter than sugar. Saccharine sodium, 500 times sweeter than sucrose. Dextrose, sucrose, sorbitol, etc. Sweetness is mainly used to inhibit bitter taste. Mannitol is mainly used in sweetening agent for chewable tablets. Next we have see about tablet coating. Purpose of coating. Tablets are coated for the following reasons: to mask the bitter taste and unpleasant odor, to improve the appearance and stability, to protect the drugs from decomposition on exposure to air and moisture in the atmosphere, to control the release of the drug from the tablet, to delay or prolong the release of drug from the tablet. That means sustained release tablet. To avoid chemical incompatibility of two drugs when formulated in the same tablet. Next move on types of coating. There are many types of tablet coating. They are sugar coating, film coating, Henry coating, and micro encapsulation, etc. First one is sugar coating. This is by pan coating method. The various stages involved in sugar coating, they are seal coating, sub coating, syrup coating, coloring and finishing. Last one is polishing. First see about seal coating. To prevent moisture penetration into the tablet core, seal coating is to be given. The material used for cell coatings are arsenic-free shellac, ethyl cellulose, or silicones. They are dissolved in a suitable solvent, and this scaling solution is sprayed on the tablets when they rotate in the pan. At least two seal coats are given. The tablets are tried between each coat. After seal coating, the tablets are removed from the pan and tried. This is the process of seal coating method. Next one is see about subcoating. Subcoating is given to the tablet to seal the surface uniform and build up tablets in a suitable size. After seal coating, the subcoating solutions is applied to the tablets and the tablets are allowed to roll. Subcoating solution is example is acacia syrup. When the tablets are uniformly wet, the subcoating powder is dusted uniformly on the tablets until the tablets roll freely. Example for this subcoating powder is talc, starch and kaolin. 3 to 4 subcoatings are usually given for the tablet. The tablets are dried between each coat. After subcoating, the tablets are dried and packed. Next one is syrup coating. The subcoated tablets are put the pan. Heavy sugar syrup is applied and the pan is allowed to rotate. 3 to 4 syrup coatings are given until tablets of suitable size are formed. The tablets are dried between each coat. Next one is coloring and finishing. In this stage, the water soluble color is dissolved in the diluted syrup. Several coatings of this colored diluted syrup are given and the tablets are dried between each coat. Then a few coatings of undiluted colored syrup are given and the tablets are dried between each coat. Finally, polishing. The dried coated tablets are transferred to a canvas lined polishing pan. Polishing solution is applied to the tablets. White beeswax or paraffin wax is dissolved in volatile solvent. This is called polishing solution. And the tablets are allowed to rotate until volatile solvent evaporates and the desired finish is obtained. These are the process of involved in sugar coating method. Next move on film coating. Film coating is a recent method of coating tablets. It has some advantages over sugar coated tablets. In film coating, the coating time is reduced. The cost of production is less, more resistant to chipping and cracking. The tablets are taken in a coating pan, film coating solutions. Example for film coating solution is polyethylene glycol, sodium carboxyl methyl cellulose, ethyl cellulose, or shellac is dissolved in volatile solvents such as chloroform or acetone. Is applied or sprayed on the tablets and the pan is allowed to rotate. If the tablets are to be colored, the dyes are dissolved in the solvent and sprayed over the tumbling tablets. Several coatings are applied and the tablets are tried between each coat. Finally, the tablets allowed to dry in warm air to form a permanent film over the tablets. Next, we discuss about Enric coating. What is mean by Enric coating? Enric coating is a type of coating that does not permit the release of drug in the acid medium of the stomach, but it increases the drug in the alkaline medium of the intestine. Yes. Enric coating is used to only release the drug in the alkaline medium of the intestine tablets. Purpose of giving an Enric coating: to prevent the decomposition of the drug in the acid medium of the stomach, to prevent the irritation of the drug in the stomach, to obtain the record action in the intestine. Material used for Enric coating is mixture of fats and fatty acid, shellac and cellulose acetophthalate. Preparation of Enric coating solution. Preparation of Enric coating solution. Cellulose acetate phthalate is dissolved in a mixture of alcohol, sorbitol monooleate, propylene glycol or acetone. When the solution is completed, the volume is adjusted with acetone. Enric coating process. The tablets are taken in a coating pan. The Enric coating solution is sprayed on the tablets and is allowed to rotate until the solvent is evaporated. Finally, the tablets are dried by means of a current of hot air. Several coatings are given until the desired coat thickness is obtained. The tablets are dried between each coat. The QC test and disintegration test for tablet. Enric coated tablets are placed in the disintegration apparatus tubes, and the tubes are allowed to move up and down motion into 0.6% hydrochloric acid for 3 hours. In this specified time, the coating particles will alone pass through the mouth, but all other portions of the tablets will retain on the mouth. Then hydrochloric acid is replaced by standard solution. Alkaline solution of pH is 6 to 8. The tubes are allowed to move up and down motion into the solution for 1 hour. In this specified time, all particles will pass through the mouth except the remaining coating fragments. Next, we see about micro encapsulation. Definition of micro encapsulation. Micro encapsulation is a process or technique by which thin coatings can be applied repeatedly to small particles of solids, droplets of liquids or dispersants, thus forming microcapsules. The particle size of the microencapsulation process is 1 by 10 of a micron to 5000 microns in size. Advantages of microencapsulation. In microencapsulation techniques, liquids can be converted into free-flowing solids, can be given coating. This technique can also design injectable ophthalmic products, suppositories, etc. The taste of bitter drugs can be masked. It is a means of formulating prolonged action dosage forms. It protects the drugs from environmental hazards. Material composition for microencapsulations. Some materials used in microencapsulations. They are core materials, coating materials, solvents, phasing out solvent means non-solvents. First, core materials. Drug either in solids or liquids. Coating material. Some coating materials is used in microencapsulations. They are methyl cellulose, ethyl cellulose, gelatin, silicones, polyacrylates, epoxy resins, gelatin, paraffin wax, cetyl alcohol, stearic acid, etc. Some solvents used in microencapsulations. The coating materials are generally dissolved in a solvent or a mixture of solvents such as ethanol, benzene, xylene, toluene, methyl ethyl ketone. Next, phasing out solvent or non-solvents. In the co-observation microencapsulation technique, the coating material is phased out by adding phasing out solvents such as polybutadiene, methyl acrylic polymer, polydimethyl siloxane, etc. Some techniques are used in microencapsulation methods. They are pan coating, fluidized bed coating, co-observation phase separation, electrostatic deposition, vacuum deposition, and air suspension coating. Evaluation of coated tablets or standard of coated tablets. Friability test, disintegration test, dissolution test, weight variation test, uniformity of diameter, content uniformity test, and hardness test. First, friability test. The friability of sugar coating is determined by trapping the tablet from a specified distance and observed whether any coating material is removed from the tablet. Next one, disintegration test. The method is similar to uncoated tablets, but the disintegration time coated tablets is 1 hour. Next, dissolution test. The method is similar to uncoated tablets. Dissolution test is indicate amount of drug dissolved in liquid. Weight variation test. This is indicate the weight differences between the tablets. Finally, today's lesson is over. What all did we learn today? Different tablets, advantages and disadvantages of tablets, classification of tablets, formulation of tablets, and types of coating. Finally, we have see about evaluation of coated tablets. Okay, thank you. See you next class.